Hi everyone, my name is Sara Biddle and I'm a PhD student with the Ichnology research group at the University of Alberta. And today I'm going to be talking about how me and my group used petrographic microfacies to reconstruct fine detailed depositional conditions of a Devonian source rock in the Northwest Territories of Canada. This Devonian source rock is the Horn River group. Over the past 2 decades, organic-rich mudstone intervals have migrated from being considered only source rocks to becoming economically viable unconventional reservoirs, as is the case for the Horn River group mudstones. As well, owing to their apparent homogeneity at the macroscopic scale or the core scale, several relatively new analytical methods are being utilized to better understand the properties of these units. This includes microfacies analysis at the petrographic level and SEM analysis at the micron scale, both of which have revealed that fine-grained successions are in fact characterized by significant lateral and vertical variation. We chose to conduct this study on the Horn River group mudstones for 2 main reasons, the first being that these units had not been previously studied at the petrographic level, and the second being that the existing interpretations of depositional settings were limited to deposition through suspension settling in distal quiet waters with anoxic to periodically eustenic bottom waters. And based on the wealth of more recent mudstone studies, this definitely needed to be looked at in more detail. The Horn River group represents middle to late Devonian deposition along the passive margin of northwestern Canada, shown in the middle diagram as the light blue Mackenzie platform. The red stars on the paleogeographic diagrams represent our study area. The Horn River group is split into 3 formations: the basal Hare Indian, an argillaceous organic-rich mudstone, a sometimes present carbonate platform and reef, the Ramparts Formation, and the upper Canol Formation, an organic-rich siliceous mudstone. The Ramparts Reef hosts the Norman Wells conventional oil field, of which the Canol Formation is the source rock. However, today we're just going to be looking at the 2 mudstone intervals of the Horn River group, so the Hare Indian Formation and the Canol Formation. The study area for this project was in the Northwest Territories, in the central Mackenzie Plain near the town of Norman Wells, about 600 kilometers northwest of the city of Yellowknife. You can see in the inset there Yellowknife outlined by the yellow star, and for those of you not familiar with the Northwest Territories, you can also see our study area outlined by the yellow box relative to Alaska on the left. Looking at the mudstones in core, you can see there isn't many macro-scale identifiable sedimentary features, and our descriptions of lithofacies were constrained to bulk descriptions of degree of facility, rock color, and the rarely identifiable sedimentary structures and macrofossils. These images here show how these mudstones differed in color and degree of facility. So you have some dark gray mudstones on the left-hand side contrasted with a lighter gray mudstone on the bottom right. You can also see some fossiliferous zones in the top right picture, as well as some pyrite alteration in the bottom right picture. But overall, not a lot of distinctive sedimentary or biogenic features at the core scale. Looking now at petrographic images of the microfacies we identified in the Horn River group, you can see there's significant heterogeneity at the sub-millimeter scale. This image represents the 8 distinct microfacies we were able to define within the Horn River group. The microfacies were defined on the basis of texture, so grain size, their mineralogic composition, any identifiable primary sedimentary structures, such as planar laminae or graded bedding, and their bioturbation intensities. Even mudstones that appear unbioturbated at the macroscopic scale or the core scale can show evidence of biogenic reworking under a microscope. So here we have 2 examples from Canol thin sections. On the left, an unbioturbated sample where biogenic reworking is estimated at less than 10%, contrasted with the image on the right showing complete biogenic homogenization, and interpreted burrows have been outlined for ease of viewing. Alright, so I'll be discussing the individual microfacies, starting with our interpreted most distal to most proximal. Microfacies 1 is the homogeneous-looking radiolarian-rich siliceous fine mudstone. It contains greater than 70% radiolarian tests, with no visible evidence for primary sedimentation mechanisms besides suspension settling, so no scour and lag surfaces or normal grading, for example, as well as it lacks significant evidence for bioturbation. The representative photomicrograph on the left shows a bed of partially dissolved radiolarians, and you can see just how abundant they are in this microfacies. Overall, the general uncompacted character of the radiolarians indicates that sediment infilling and recrystallizations of the test to chert or calcite occurred prior to compaction, which is consistent with formation during early diagenesis. and the absence of significant bioturbation and this inferred early diagenetic cementation of the radiolarian tests indicates that the microfacies formed in still bottom waters with relatively low dissolved oxygen concentrations, likely from suspension settling of proliferation pulses of these radiolarians. Our second microfacies is the homogeneous-looking dolomitized argillaceous fine mudstone. It contains 20 to 90% diagenetic dolomitization, and the dolomite is mostly rhombic, but can also be microcrystalline or a replacement of shells. And unfortunately, any primary sedimentary structures or bioturbation has been overprinted by the dolomite, so we weren't able to identify any. Our photomicrograph here shows the pervasive Faroan nature of most of the dolomite within this microfacies. So overall, diagenetic dolomite, both as rhombs and microcrystals, is associated with post-depositional alteration of organic-rich mudstones through microbial sulfate reduction at or near the sediment surface. And when it forms in early diagenesis, it's considered to be an anoxic mineral that forms under strongly reducing conditions. Therefore, these pervasively dolomitized sediments are interpreted to represent slow sediment accumulation rates under poorly oxygenated regimes, combined with long residence times at the seafloor, all of which are conditions that permit the byproducts of anaerobic bacterial reactions to accumulate and form this early diagenetic Faroan dolomite. Microfacies 3 is the discontinuous wavy-parallel laminated to homogeneous-looking argillaceous fine mudstone. This fine mudstone microfacies contains only rare and sporadically distributed fine silt grains, so making up less than 5% of the matrix. This likely indicates deposition near the end of a sediment transport path. Similar to the detrital silt grains, radiolarian and conodont microfossils occur dispersed throughout the sediment as opposed to being concentrated along specific bedding planes. And so we can see that in the photomicrograph here, where you have 3 radiolarians dispersed throughout the matrix, almost floating throughout the clay matrix. The organic matter in this microfacies is interpreted to be phytodetritus, so aggregates of organic matter, detrital sediment, and microorganism tests, and it's interpreted to be delivered to the sediment surface through suspension settling. Biogenic pelagic elements such as radiolarians, conodonts, and carbonate fossils indicate the presence of an overlying oxygenated water column, whereas the rare and difficult to see microburrows suggest partial oxygenation of the sediments. And so overall, this microfacies is interpreted to be the result of hemipelagic suspension settling under poorly oxygenated bottom waters. Microfacies 4 and 5 are compositionally identical, they just differ on the intensity of bioturbation. So microfacies 4 is the rarely bioturbated, discontinuous, wavy-parallel laminated, silt-bearing fine mudstone, and microfacies 5 is the bioturbated equivalent. Overall, microfacies 4 has bioturbation in the range from 0 to 10%, and microfacies 5 ranges from 10% to 100% bioturbated. These two microfacies have a detrital silt content of less than 30%. The photomicrograph on the side here shows the bioturbated version, so microfacies 5, with the interpreted burrows outlined in the bottom left-hand corner. The increase in detrital silt content is interpreted to indicate increased proximity to the sediment input source or an increase in flow competency associated with a steepening basin floor gradient. Microfacies 4 and 5 occasionally occur as discrete homogeneous-looking beds and bed sets in other microfacies, interpreted as storm deposits. Submillimeter stacked normally graded beds can be seen in some cases, finding upwards from phytodetritus and interclast-rich bases to homogenized clay tops, indicating deposition from waning flow. The bioturbated version is interpreted to be the more proximal expression of microfacies 4. Enhanced water column mixing from strong bottom currents may have facilitated more intense biogenic reworking due to increased concentrations of dissolved oxygen brought in by the flows when compared to the flow strength interpreted for microfacies 4. Stronger bottom currents and their associated carrying capacity are interpreted to stem from increased proximity to the shoreline and enhanced storm influence compared to the rarely bioturbated, more distal microfacies. Overall, these microfacies are interpreted to represent combined suspension settling and bedload transport in more proximal settings with fluctuating bottom water oxygenation. In this photomicrograph, we have an example from microfacies 4, the rarely bioturbated version. We have 3 discontinuous silt laminae where they have punctuations laterally throughout, Those punctuations are interpreted to represent disruptions by bioturbating organisms, but overall, the silt laminae are interpreted to represent evidence of bed load transport of the silt grains. Microfacies 6 is the bioturbated discontinuous planar parallel to continuous wavy non-parallel laminated argillaceous siliceous medium mudstones. It's the coarsest grained of all the microfacies we identified, containing greater than 30% detrital silt. The increase in detrital silt content reflects deposition in a proximal, relatively high energy setting when compared to the previously described microfacies. Although a large fraction of the detrital volume does occur as clay, it occurs as clay aggregate grains. Clay aggregate grains are silt-sized accumulations of clay platelets, and they behave hydrodynamically similar to detrital quartz silt. The SEM image here shows these aggregate grains, outlined in yellow, within a silt-rich section from the Canol Formation. The predominance of these clay aggregate grains indicates that deposition occurred under heightened depositional energy and sediment input, likely from turbid waters or resuspension of semi-consolidated muds. Here we have a zoomed-in image of one of these clay aggregate grains, bordered by 3 detrital quartz silt grains, so you can see how the look and shape of these clay grains is very similar to the detrital quartz silt. Microfacies 6 also contains several primary sedimentation structures that indicate relatively energetic deposition. So here we have an example of unidirectional current ripples identified in one of the Horn River group samples. As well, we have distinct wave-enhanced sediment gravity flows, which are gravity-driven flows where the energy is provided by agitation from surface waves. In this sample, you can see the distinctive 3-part layering, starting with erosion and deposition in bed A, then planar laminae from the laminar gravity flow in bed B, and finally the homogeneous appearing fine-grained deposit from the waning end of the flow in bed C. Overall, microfacies 6 is interpreted to represent bed load traction transport under oxygenated bottom waters. Microfacies 7 is the fossiliferous, discontinuous to continuous wavy-parallel laminated argillaceous fine mudstone. It contains greater than 30% tentaculated tests and has 2 main appearances. The first, shown in the photo here, is intact tentaculatids dispersed randomly throughout a clay matrix, and it's commonly dolomitized. This occurrence is interpreted to represent suspension fallout of tentaculatids, with the dolomitized interiors indicating pre-compaction alteration, indicating long residence times between sedimentation events. And here we have a zoomed-in image showing the pre-compactionally dolomitized interiors of the tentaculatids. The second phase of the fossiliferous microfacies is made up of relatively thick accumulations of fragmented and undolomitized tests, intercalated with clay-dominated beds. These are interpreted as winnowed shell lags, where the winnowing was initiated by storm-generated bottom currents. The intercalated clay beds represent returns to fair weather sediment conditions. Overall, the fossiliferous microfacies is interpreted to represent suspension settling of tentaculated tests into a location subject to periodic storm influence. The final microfacies we identified was the interclastic-rich, discontinuous, planar parallel laminated, argillaceous fine mudstone. It's characterized by greater than 30% volume of interclastic aggregates. These aggregates are the lenticular lighter-colored features you see in the image here, and they're interpreted as the product of up-dip erosion of previously deposited semi-consolidated muds that have been transported to more distal locations and subsequently flattened through compaction. These interclasts can be commonly misinterpreted as pelagic fecal pellets or cross-sections of small microburrows. Here we have an SEM image from the Canol showing these interclasts, and they're outlined here by the white dotted lines. In this image, we're seeing how they're compositionally identical to the surrounding matrix, with the exception of having less pyrite in them, so less of those bright spots, and less organic matter, represented by the black areas. If these features were pelagic fecal pellets, you'd expect them to contain abundant pelagic tests, such as coccoliths. And here we have a bedding parallel thin section showing the distinct margins of the interclasts. If these were burrow features, like some have interpreted, you'd expect to see them as tube-like features crossing the entire thin section, not isolated bodies. Overall, microfacies 8 is interpreted as the most proximal microfacies, resulting from constant basinward transport of suspended clay sediment in turbulent or laminar flow, with coarser grain components such as silt and sand-sized interclasts being transported as bed load. a general absence of incorporated organic matter and pyrite rhamboids in the interclasts is interpreted to reflect persistent bottom current energy that supplied a constant source of interclasts and clay floccules for more proximal oxygenated settings, where the well-oxygenated waters did not allow for the accumulation of organic matter and authigenic pyrite in the sediments. These interclast-rich beds can also occur as stacked graded beds, finding upwards from interclast-rich bases to clay drapes at the top, each representing distinct storm events. So, if we were to envision a hypothetical pro-grading depositional profile for the Horn River Group mudstones, it would go something along the lines of this. First, suspension settling and accumulation of clay floccules, organic detritus, and microfossils, leading to the formation of our clay-dominated microfacies, or microfacies 3. Next, a proliferation event might result in the accumulation of abundant radiolarians, resulting in our radiolarian-rich mudstone. Next, we're getting a little proximal, so we have more silt coming in, so deposition of silt, clay, and still organic matter. So we get accumulation of our silt-rich mudstones. Next, an increase in silt size, as well as an increase in depositional energy, leads to the formation of microfacies 6, which is our silt-rich microfacies. And here we see our wave-enhanced sediment gravity flows more proximal and our current ripples down dip. Then we move on to microfacies 7, where we get our tentaculatids being deposited. So in more distal locations, they have that suspension settled appearance where they have well-preserved interiors and they're heavily dolomitized. In more proximal locations, the tentaculatid-rich beds have been winnowed and accumulated into shell lags. And then finally, our clastic sediment input source is approaching our location, and so we get a big influx of our clay and our up-dip eroded intraclastic aggregates, leading to the formation of our intraclastic-rich mudstone. So, in summary, the Canol and Hare Indian mudstones, which were previously thought to represent quiescent suspension settling environments under poorly oxygenated bottom waters, actually represent dynamic depositional settings with a combination of bed load and suspension settling mechanisms, as well as persistent anoxia is unlikely due to the evidence we have for biogenic reworking. We have many people to thank for the outcomes of this study, namely the Northwest Territories Geological Survey for funding the project, and Husky Energy, ConocoPhillips, and Paramount Resources for giving us access to the core and thin section datasets. Thanks everyone for listening to my talk, and if you're interested in any more details on the subject, they're all available in our recently published paper in Sedimentology.